I want you to picture something for me. You're lying on an operating table. You are, believe it or not, fully conscious.
A little weird to think about.
Yeah. You're comfortable, maybe a little groggy from the medication, but you are wide awake. You're actually, chatting with a doctor.
But here's the kicker. The really wild part of the situation.
The top of your skull has been removed.
Mm-hmm.
The air in the room, the sterile recycled air of the OR, is literally touching the surface of your brain.
It's such a visceral image, isn't it? But for a patient named Bev, who we read about in our source today, and for thousands of others, this wasn't some scene from a horror movie.
Not at all.
It was, a Tuesday. This comes directly from our source material, Behavioral Neuroscience, 8th edition.
Right. This is the story of Bev. She's undergoing brain surgery, and the surgeon is the absolute legend, Wilder Penfield.
A giant in the field.
And he's standing over her with this tiny electrode. It was basically a live wire with a very, very gentle current. And he starts touching the surface of her brain with it.
And this is where that whole idea of the ghost in the machine really gets put to the test. Because when Penfield touches the brain, Bev doesn't feel any pain.
Which is wild in itself.
It is. The brain itself has no pain receptors. It can process pain signals from everywhere else, but it can't feel itself being touched. It's a fascinating sort of evolutionary quirk.
So instead of pain, what does she report?
Experiences.
Yeah, that's the part that just it blew my mind reading it. He touches one specific spot, and she feels a tingling in her hand. Nothing's touching her hand, but she feels it.
The sensation is generated directly in the brain. He moves the electrode just a few millimeters, stimulates again.
And bam, she sees flashes of blue light.
He moves it again, just a tiny bit, and suddenly she's not in the operating room anymore. She vividly smells the scent of a childhood haunt, a place she hasn't thought about in decades.
Or she hears a fragment of a song, crystal clear, as if it's playing on a speaker right there in the room.
Yeah.
But it isn't.
It forces us to confront a really, let's say, an uncomfortable truth. We like to think of our memories, our personality, our soul as these ethereal, intangible things.
Sure, yeah. The ghost.
Exactly. But Penfield basically proved that if you poke the hardware, you change the software, you touch a piece of, well, gray jelly.
And a specific memory just pops out.
It's unbelievable. And this procedure, cortical electrical stimulation mapping, it wasn't just for research. This was a clinical necessity. Penfield was trying to find and remove diseased tissue that was causing Bev's seizures.
Right, but he had to do it without damaging key functions.
Right.
Like her ability to speak or move her hand.
So he had to map it first. He had to ask the brain, hey, what do you do before he cut?
It sounds like magic. I mean, it really does. But as we're going to learn today, it isn't magic. It's biology.
It's engineering. It's the hardware of human behavior.
Welcome to the deep dive.
Today, we're unpacking what might be the most complicated object in the known universe. That 3-pound organ sitting right behind your eyes.
We have a massive stack of research today from Breedlove and Watson's text. And the mission is, well, it's pretty simple. We're going to tear that hardware apart to see how it works.
We're not just looking at pictures.
No, we want to understand the mechanics.
And we're going to do this the way neuroscientists do, by following the biological hierarchy. Strictly. We're not going to skip any steps. We're going to start with the chassis, the physical structure of the cells.
The building blocks.
Then we'll look at the battery, the electrical sparks that carry all the messages. After that, the fuel injection system, so to speak. The chemicals that bridge the gaps between cells.
And finally,
the climate control. The hormones that signal these big, slow, body-wide changes.
So we're literally building the machine from the ground up. We're going from a single microscopic cell all the way up to the feeling of a social connection.
Precisely. And just a quick disclaimer before we really dive in. We are sticking strictly to the facts laid out in this text.
That's right.
No outside theories, no wild speculation. Just the foundational science from Breedlove and Watson that explains how you're able to hear my voice and understand it right now.
Okay, let's do it. Let's unpack this thing. Section 1. The architecture. Functional neuroanatomy.
Right. So to understand the machine, you have to understand the unit it's built from, the fundamental component. But interestingly, for a long, long time, scientists couldn't even agree on what that unit was.
That seems like a pretty big hurdle.
A huge one. Back in the 19th century, before we had things like electron microscopes, looking at brain tissue was just really confusing.
I can imagine. If you look at a slice of liver or a kidney under a microscope, you see these nice, neat little bricks, right? You can see the distinct cells.
Exactly. You can see the borders. But the brain,
Just a mess.
it just looked like a tangled mess.
Like a bowl of spaghetti where you can't tell where one noodle ends and the other begins.
That is a perfect analogy. And this visual mess led to this massive scientific feud. We'll call it the soup versus spark debate later on, but initially it was just about the physical structure.
Who are the main players in this fight?
On one side, you had the Italian scientist Camillo Golgi, a very respected figure. He championed what was called the reticular theory.
Reticular.
Yeah.
Like a reticulum or a net.
Exactly. He believed the brain was a continuous network of tubes, a mesh. He thought all the cells were physically fused together like a giant, intricate plumbing system.
So information would just flow like water through pipes.
Yes, continuously, without any interruption. And to be fair to Golgi, if you just squint at a slice of brain tissue, that's kind of what it looks like. It makes intuitive sense.
It does. It looks like one big connected thing.
It does. But then enters the hero of this section, Santiago Ramón y Cajal, a Spanish neuroscientist who was also, and this is important, a brilliant artist.
An artist.
Yes. His drawings were incredible. Now, the irony here is just delicious. I love this part of scientific history because it's just so wonderfully petty.
Oh, this sounds good.
Cajal actually used a staining technique invented by Golgi himself to prove Golgi wrong.
Oh, no way. That is the ultimate scientific power move.
Isn't it? I'm going to take your invention, your life's work, and I'm going to use it to just completely dismantle your central theory.
So what was so special about this Golgi stain?
The Golgi stain is unique because, for reasons we still don't fully understand, it only stains a tiny percentage of neurons black against a light background, maybe 1%, sometimes less.
So it pulls a few noodles out of the spaghetti bowl so you can actually see them.
A perfect way to put it. It allows you to see the individual shape of a single cell in complete isolation, without the visual clutter of the billions of cells around it.
And Cajal looked at these silhouettes.
He looked at these beautiful, stark silhouettes, and he realized something profound.
They weren't fused together.
They weren't fused. He created these incredible pen and ink drawings, which are featured in our source material. They're really works of art. They showed neurons as distinct individual shapes. They were touching, or almost touching, but they were separate entities.
He said they were contiguous, not continuous.
Exactly. They were kissing, not merged. And this observation, the simple fact, led to what we now call the neuron doctrine.
And this is what, like the central dogma of neuroscience?
It is the absolute bedrock of modern neuroscience. It has two main rules. First, the brain is composed of separate cells that are structurally, metabolically, and functionally independent.
Yeah, they're individual units.
Right. And second, and this is the absolute game changer, information is transmitted from one neuron to the next across tiny gaps.
Those gaps are the synapses.
It's the synapses, and this is so crucial for you to understand. If Golgi was right, if the brain was just a fused pipe, the signal would just flow like water. You couldn't stop it. You couldn't modify it. It would just go.
There's no control.
No control. But because there's a gap,
the signal has to make a jump.
it has to jump. And that jump provides the opportunity for control. You can block the jump. You can speed it up. You can slow it down. That tiny gap is where learning happens. It's where decision-making happens. It's where the entire complexity of the human mind comes from.
It's amazing that such a huge concept hinges on such a tiny, tiny space.
The entire game is played in that space.
Let's talk about the scale of this, because the numbers that the text throws out are honestly hard to wrap my head around.
They are astronomical. I mean, truly, the adult human brain has somewhere in the neighborhood of 80 to 90 billion neurons.
Billion with a B.
With a B. But the neurons are just the players. The synapses, those gaps, are the conversations. Estimates vary, but we are looking at something like a quadrillion synapses.
A quadrillion? What even is that?
It's 10 to the power of 15, a 1 with 15 zeros after it.
I can't even visualize that. Is there an analogy?
The text provides a great one. Imagine a single grain of sand represents one synapse, just one tiny connection. If you gathered a quadrillion grains of sand, they would fill a cube larger than an American football field. We're talking 1 million cubic yards of sand.
That's staggering. That is the level of connectivity we are dealing with inside our own skulls.
It is. And navigating that football field of sand, we have these very specific cells, the neurons. And the text does a great job of breaking down the neuron into 4 specific zones. This helps us understand that a neuron isn't just a blob, it's a machine with distinct parts.
Right, it's an assembly line.
Yeah.
Information comes in one end and goes out the other.
Exactly. And no matter how weird a neuron looks, and they come in some amazing shapes, from pyramids to stars to these things called Purkinje cells that look like giant sea fans, they almost all have these 4 functional zones.
Okay, what's the first one?
First, you have the input zone. These are primarily the dendrites.
The receiving antennas.
Yes. They branch out from the cell body like the branches of a tree covered in little spines, all to catch signals from other neurons.
So they collect all the incoming mail.
They do. Then you have the integration zone. This is usually the cell body or the soma. This is where the nucleus lives, where the cell's DNA is. But functionally, its job is to combine all those inputs from the dendrites.
So it reads all the mail and decides what to do.
Precisely. The neuron has to make a choice here. Do I fire or not? That all-important decision happens in the integration zone, specifically at a spot called the axon hillock.
Okay, so the decision is made. Go. Then what?
Then the signal has to travel, sometimes a very long way. That happens in the conduction zone. This is the axon. Think of it as the wire. It's a single, slender extension that carries the electrical impulse away from the cell body toward its target.
And it can be really long, right? Like from your spine down to your big toe.
Incredibly long, a single cell. And finally, at the end of that wire, it has to deliver the package.
The output zone.
The output zone. These are the axon terminals, or synaptic boutons. This is where the signal gets passed to the next cell. This is where the electrical signal gets converted into a chemical signal to cross that synaptic gap we talked about.
Input, integration, conduction, output. A one-way street for information.
Always. Information doesn't flow backward in a healthy neuron.
Now, we also have to give a quick shout out to the support staff. The neurons get all the glory, but the text makes a point to mention glial cells.
The forgotten heroes of the brain. Glia literally means glue in Greek.
So we thought they just held everything together.
For a long, long time, yes. We thought they were just like packing peanuts, providing structural support. But we now know they do so much more. They provide nutrients, they clean up waste. Some of them form the myelin sheath that insulates axons.
They're like the pit crew for the race car.
That's a great way to put it. They are essential. Without them, the neurons can't function and will die.
Speaking of seeing these structures, the text shows us how far we've come from Cadell's beautiful pen and ink drawings. We can now map these connections in living, breathing humans using something called DTI.
Diffusion tensor imaging. Yeah, this is a fascinating application of physics, of MRI technology. It tracks the movement of water molecules in the brain.
Wait, hold on. How does watching water tell us anything about brain wiring? That seems like a stretch.
It sounds it, but it's actually quite clever. Think about a bundle of straws, a whole box of them.
Okay.
If you pour water into that box, the water can move very easily up and down inside the straws, right, along their length.
Sure.
But it can't really move sideways through the plastic walls of the straws.
Right, it's contained.
The axons, those conduction wires, are like microscopic straws. Water diffuses very easily along the long axis of the axon, but it's blocked from moving sideways by the fatty myelin sheath that covers them. The official term for this tendency is fractional anisotropy.
That is a mouthful.
It is, but the concept is simple. The DTI scanner detects the direction in which water is moving most easily, and by doing that, a computer can draw lines representing the white matter tracks, the major cables of the brain.
So you get a wiring diagram.
You get a wiring diagram. It's called tractography. These incredible multicolored images that show us exactly which parts of the brain are hardwired to which other parts. It's like having the city's subway map.
And the text takes this one step further. It's not just about mapping a single brain anymore. There's this concept of social neuroscience and this incredible image of a dyadic functional MRI.
This is really the cutting edge. Typically, an MRI is one person alone in a dark tube. It's very isolating.
Sure.
But this setup has 2 scanners, 2 people, and they can look at each other through a series of mirrors and screens. They can interact. So we're starting to map not just the biology of a single isolated organ, but the biology of the interaction between 2 organs.
We can watch 2 brains talking to each other in real time.
In a sense, yes, we're mapping the hardware of connection.
Which is a perfect segue. We've built the map, we know the structure, we know the parts of the machine, now we need to turn the power on. This brings us to section 2, the spark, neurophysiology.
This is where we get into the how. How does a thought, an impulse, travel from one side of that football field of sand to the other? And the core rule you need to remember is this. Information within a neuron is electrical. Information between neurons is chemical.
So inside the cell, it's a spark. At the gap, it's a squirt of chemical.
Precisely. Let's focus on that spark first. The text uses a battery analogy, which is good, but I want to be really clear. It's not electricity like the copper wire in your wall.
It's not that fast.
It's not that fast, no. The signal doesn't travel at the speed of light. It's actually surprisingly slow, all things considered.
Because it's biological.
Because it's chemical electricity. It's based on the movement of ions. So think of a neuron that's just sitting there at rest, not firing. It's not actually doing nothing. It is furiously working to act like a battery. It maintains a membrane potential. It stores a charge.
It's building up potential energy.
Exactly. And it does this by manipulating ions charged particles. The key player here, the absolute star of the show, is sodium, the ion Na plus A.
Okay, sodium.
The neuron has these tiny molecular pumps, and they are constantly working, burning energy to pump sodium out of the cell. It's actively kicking it out the door.
So you end up with a lot of sodium on the outside and not very much on the inside.
Correct. And this creates a massive pressure differential, a huge gradient. The text details two forces at work here. First, there's diffusion.
Things moving from high concentration to low.
Right, like a drop of food coloring spreading out in a glass of water. The sodium is really crowded outside the cell, so it desperately wants to get inside where it's less crowded.
Okay, that's one force pushing it in.
The second force is electrostatic pressure, or, electrostatic force.
Yeah.
Opposites attract, likes repel.
The basic rule of magnetism.
Yes, the inside of the neuron at rest is negatively charged. Sodium ions are positively charged. So like a magnet, the negative inside is pulling that positive sodium in.
So let me get this straight. You have the force of diffusion pushing sodium in, and you have the electrical force pulling it in. It's getting hit with a double whammy. Why doesn't it just rush in?
Because the doors are locked. The cell membrane is selectively permeable. It has channels or gates. And the ones for sodium are closed when the cell is at rest. It keeps the sodium out. So the neuron is like a drawn bowstring. It is under incredible tension. It is holding back this massive river of sodium that is absolutely desperate to get inside.
The dam holding back the river. That's a great image.
It's exactly that. So when the fire signal comes, when the neuron gets excited, it doesn't have to generate the energy to send the signal. The energy is already stored. It just has to open the gates.
Unlock the doors.
And the river floods in. The sodium explodes inward, and that sudden, massive rush of positive charge flips the voltage of the cell from negative to positive in a fraction of a millisecond.
And that flip is the signal.
That flip is the action potential. It's the spike. It's the spark. And it's a wave that sweeps down the axon like a burning fuse on a firecracker.
And the text makes a really important point that this is an all-or-nothing event. You can't have a half-action potential.
Right. It's like firing a gun. You either pull the trigger hard enough to fire the bullet, or you don't. You can't kind of fire a bullet. Once the neuron hits its threshold, it fires, and that spike travels with the same intensity all the way to the end of the axon.
Okay, but wait. If the neuron just fires or doesn't fire, if it's just binary, on or off, how do we process complex information? How do we decide to eat a sandwich versus writing a poem? It can't all just be on or off.
That's a fantastic question, and the answer happens back in the integration zone. Remember, a single neuron is getting inputs from thousands, sometimes hundreds of thousands, of other cells.
It's not just one guy telling it to fire.
No, it's a whole chorus. It's a committee meeting.
Okay.
And some of those inputs are excitatory. They're saying, fire, fire. These are called excitatory postsynaptic potentials, or EPSPs. They cause a small local depolarization, trying to push the neuron closer to that firing threshold.
They're opening a few of the sodium doors, just a crack.
A good way to think of it. But other inputs are inhibitory. They're saying, don't fire, stay quiet. These are inhibitory postsynaptic potentials, or IPSPs. They do the opposite. They make the inside of the cell even more negative, pushing it further away from the threshold.
So it's basically a voting system.
It is exactly a voting system. The cell body at that axon hillock is constantly adding and subtracting these little potentials. It's summing up the votes. And if the yes votes, the EPSPs, outnumber the no votes, the IPSPs, enough to push the membrane potential to that critical threshold,
Action potential.
it fires. If not, it stays silent. Every single thought you have, every movement you make, every sensation you feel is the result of billions upon billions of these tiny elections happening every single millisecond.
That's incredible to think about. It's democracy at a cellular level.
It is.
But like any complex electrical system, it can short circuit.
And the text brings us back to the story of Bev and another patient named Deidre to discuss epilepsy. Right. Epilepsy is at its core an electrical storm in the brain. The text describes a grand mal seizure, which is an older term. The more accurate name now is a tonic-clonic seizure.
So what's actually happening electrically during one of these seizures? Why does the person lose control?
Normally, brain activity is desynchronized. It's chaotic. Different parts are doing different things. You're processing vision, hearing, memory, planning your next sentence. On an EEG, which measures brainwaves, it looks like noisy static.
Right.
But during a seizure, a huge population of neurons starts firing in high-frequency synchronized bursts. They all start doing the same thing at the same time.
It's like an entire stadium of people all shouting the exact same word at the exact same time.
An excellent analogy. And this wave of synchronization sweeps across the brain. The text shows an EEG of a seizure, and instead of chaotic noise, it looks like these giant, rhythmic, spiky waves. This massive electrical discharge completely overwhelms the normal function of the system, causing the convulsions and the loss of consciousness.
And going back to Deidre, the patient mentioned in the book, the text says that during her stimulation mapping, like Bev's, the doctor stimulated one spot and she just found it funny. She started laughing.
Right. Without a joke. Without any context. The electrical pulse alone triggered the feeling of amusement.
That is just spooky.
It is, but it's also profound. It shows that even something as abstract and fundamentally human as a sense of humor is, at its core, an electrical pattern in a specific part of the machine. If you stimulate the humor circuit, you feel amusement. The feeling is a direct consequence of the hardware's activity.
Now, speaking of stimulating circuits, the text introduces a technology that sounds like it's straight up from the future. Optogenetics.
This is an absolute game changer in neuroscience research. With Penfield, we use electricity to stimulate the brain in humans, but it's a bit crude. It stimulates everything in the area.
Like hitting a button with a boxing glove.
That's a great way to put it. Optogenetics allows us to control specific types of neurons with the precision of a laser beam using light.
But how do you get a neuron to respond to light? They're buried inside a skull, in the dark.
You have to borrow a trick from algae.
From algae.
Yep. There are certain algae that float in the ocean and need to find sunlight to photosynthesize. They have these special proteins in their membranes called channel rhodopsins, and these proteins are basically light-activated gates.
So they're doors that open when light hits them.
Exactly. When blue light hits this protein, the channel opens and lets positive ions flow in. So scientists brilliantly figured out how to take the DNA that codes for that protein and, using a harmless, modified virus, insert it into specific neurons in a lab animal like a mouse.
So you could target, say, only the neurons that produce dopamine.
Precisely. So now the mouse has dopamine neurons that have these little light-activated switches embedded in them.
And then you just shine a light on them.
Pretty much. The text shows a diagram of a mouse with a tiny, flexible fiber optic cable surgically implanted into its head, leading down to that specific brain region. When the researchers turn on a blue laser,
the channels open, sodium rushes in, and those specific neurons fire.
Instantly. It's like mind control via flashlight.
Wow.
It allows us to prove with absolute certainty which neurons drive which behaviors in a way we never could before. We can literally turn a specific behavior on and off like a light switch. It's one of the most powerful tools we have.
Okay, so we have the structure and we have the electrical spark. But remember the problem we talked about earlier, the gap. The spark hits a dead end at the axon terminal. It can't just jump across that gap to the next cell.
It can't. The electricity hits the synaptic cleft, which is filled with fluid, and this is the fundamental problem of the brain. Electricity cannot jump through salt water very well, not across a gap like that, without losing its integrity. The spark is well and truly stuck.
So the entire system has to change modes. It has to translate the message from one language to another. This is section 3, the chemistry. Psychopharmacology.
This is the critical transition from spark back to soup. When that electrical action potential, that wave of positive charge, hits the very end of the line, the axon terminal, it changes the game. It doesn't try to jump. Instead, it triggers the opening of a different kind of channel. Voltage-gated calcium channels.
Calcium, I always think of that for bones and teeth.
Here, it acts as a crucial signal flare. When these channels open, calcium ions, which are much more concentrated outside the cell, rush into the terminal.
And what does the calcium do?
The influx of calcium is the trigger that causes these tiny bubbles inside the cell, they're called synaptic vesicles, to move towards the edge of the terminal. And these vesicles are filled with the chemical cargo, the neurotransmitters.
So the electricity's only job at the end is to let the calcium in, and the calcium is what actually releases the chemicals.
Precisely. The calcium causes these vesicles to dock with the outer membrane and then burst open, a process called exocytosis. Think of it like a tiny ferry boat docking and lowering its ramp. The vesicles fuse with the membrane and spill their chemical contents out into the synaptic cleft.
So the electrical signal is gone. It died to the terminal, and now we have these chemical molecules just floating across the water-filled gap.
Right. They drift across that tiny space and bump into the input zone, the dendrites, of the next cell. But they don't just enter the cell, they work like a key in a lock. They bind specific protein structures called receptors.
And this key and lock analogy is vital for anyone interested in medicine or, how drugs work. Because the text defines a ligand as basically anything that binds to a receptor. That includes the brain's natural neurotransmitter.
And it includes drugs. Most of the drugs that affect our brain and behavior are receptor ligands. They are essentially counterfeit keys that have been designed to fit into those same biological locks.
So if I take a drug, I'm basically just flooding my brain system with a bunch of fake keys.
Exactly. And those keys can do different things. Some drugs are agonists. They're like a good counterfeit key. They fit in the lock and they turn it, opening the door and mimicking the effect of the natural chemical.
Okay. And the opposite.
The opposite is an antagonist. That's like a key that fits in the lock but is broken off. Doesn't turn, it doesn't open the door, it just jams up the lock so the real natural key can't get in and do its job.
The text uses serotonin as a prime example of how this whole chemical system is organized. It shows this amazing diagram of the serotonergic pathways.
It's a fascinating image. It's a side view of the brain, a sagittal view. And you can see that the source of almost all the serotonin in the brain is this tiny, tiny cluster of cells deep in the brainstem called the Raphi nuclei.
It looks so insignificant on the diagram.
It really does. But from that tiny source, those neurons send their axons out and they spray across the entire cortex. The frontal lobe, the temporal lobe, the parietal lobe, everywhere. It's like a sprinkler system.
So a very, very small number of cells can have a massive influence on the mood and function of the entire brain.
Exactly. It's an incredibly efficient system. And that's why drugs that target serotonin, like many common antidepressants, the SSRIs, can have such widespread effects on things like depression, anxiety, sleep, appetite. They are tweaking the chemical soup that the whole brain is swimming in.
But we can't talk about drugs without talking about the dark side of this chemical hacking, addiction. The source material details an experiment that I think is one of the most famous and maybe one of the starkest in all of psychology, the self-administration setup.
This is how we measure reward scientifically, how we quantify the addictive potential of a substance. You take a lab rat and put it in a box with a lever, and it's connected to a system where, when it presses the lever, a pump injects a tiny dose of a drug directly into its veins or into its brain.
And the result is pretty telling, isn't it?
It's chilling. If the drug is saline, just salt water, the rat presses the lever a few times out of curiosity, realizes it does nothing, and then completely ignores it. But if the drug is something like cocaine or methamphetamine,
The rat just keeps pressing it.
it will press that lever thousands of times.
Thousands for a single dose.
Thousands of times. They will ignore food, they will ignore water, they will ignore a potential mate. They will press the lever until they collapse from sheer exhaustion.
That is terrifying because it shows it's not a moral failing on the rat's part. It's not about willpower. It's a hardware hack.
Precisely. It demonstrates that these drugs tap into and hijack the brain's fundamental reward system, the circuitry that evolved to encourage survival behaviors like eating and reproducing, and they hijack it completely. The drug becomes the only survival signal that matters to the brain.
But the text also mentions a potential solution that is equally fascinating and purely biological: vaccines for addiction.
This is a really clever outside-the-box approach. The diagram in the text shows how it works. A vaccine is created that stimulates the body's immune system to create antibodies that specifically target drug molecules, say cocaine molecules.
So your body starts to treat the drug like it's a virus or a bacteria.
Exactly. The antibodies circulate in the blood, and when the person uses the drug, the antibodies bind to the drug molecules. This creates this big antibody-drug clump that is physically too large to cross the blood-brain barrier.
It's a biological firewall.
It is. The drug is in the person's body, but it can't get into the brain to hit those receptors in the reward circuit. So the user would take the drug and feel nothing. No high, no reward.
And that would, in theory, break the cycle of the lever pressing.
Theoretically, yes. It's a fascinating idea. It treats the chemistry of the addiction by simply blocking the drug's access to the hardware.
Okay, so we've gone from electricity to chemistry. We've seen the spark in the soup happening at the speed of milliseconds.
Hmm.
But there's one more layer of communication in the body, a slower, more rhythmic, system-wide signal regulation.
Right.
Section 4, the body-wide signal.
Hormones.
We're definitely shifting gears here. We're moving from the time scale of milliseconds, the speed of a neuron firing, to a time scale of hours, days, or even entire lifetimes. Hormones are the body's long-distance bulk messaging system. The text starts with a bit of a history lesson here, which I loved, going all the way back to the ancient Greeks and their concept of the humors.
It's important to realize that long before we knew about neurons or synapses, we knew about fluids. We understood that things in our blood affected how we felt and acted. The Greeks believed personality was determined by the balance of 4 bodily fluids: phlegm, blood, black bile, and yellow bile.
And it's amazing how we still use words derived from this today. It's baked into our language.
We absolutely do. If someone is phlegmatic, we mean they are sluggish and unemotional like phlegm. If they are sanguine, from sanguis, the Latin for blood, they're cheerful and optimistic.
Bilious means irritable. Choleric means hot-tempered, from yellow bile or choler.
It's amazing how language preserves that old biology. We had this deep intuitive understanding that chemicals in our body changed how we acted, even if the specifics were completely wrong.
But the real science of hormones, of endocrinology, started with a very weird and, frankly, a kind of brutal experiment involving roosters.
Yes, Bertholdt's experiment in the 19th century. This is the classic knockout and replacement study, but done with a scalpel instead of genetics.
Walk us through what he did.
So Berthold took groups of young roosters. In one group, he did nothing. They were the control. In another group, he castrated them. He surgically removed their testes.
Okay, and what happened to those birds?
They grew up looking and acting very different. They had small combs and wattles, they didn't crow, they weren't aggressive towards other males, and they showed no interest in hens. They didn't develop normal rooster behaviors.
Then he did the twist, the replacement part of the study.
He did. In a third group of roosters, he removed the testes, but then he took one of the testes and he implanted it back into the rooster's abdominal cavity, into its belly. Now, critically, he didn't reconnect the nerves. There was no wiring. The organ just sat there and eventually established a blood supply.
So if the signal that makes a rooster was nervous, if it traveled through a wire, this shouldn't have worked.
It shouldn't have worked at all, but it did. That rooster developed normally. It grew a big comb, it crowed loudly, it fought other roosters. This was the elegant proof. It showed that the signal wasn't nervous. It was a chemical released by the testes into the blood, which then traveled throughout the body and to the brain to change development and behavior.
And that chemical was testosterone.
That chemical was testosterone. It's a beautifully simple, foundational experiment. The signal is in the blood.
And that leads us directly to modern behavioral endocrinology. We now look at how these blood-borne signals affect incredibly complex social behaviors. And the text highlights a really powerful study on the hormone oxytocin and its connection to autism spectrum disorder, or ASD.
Right. ASD is, in part, defined by challenges with social interaction and difficulty judging the thoughts or intentions of others. So the natural question for a neuroscientist is there a biological reason for this? Can a hormone maybe treat it?
And they used a mouse model to test this.
They did. They had knockout mice that were missing a specific gene related to social behavior, which resulted in them showing social deficits similar to what we see in ASD. They put these mice in a test chamber with 3 compartments.
Okay.
On one side, there was an empty room. On the other side, a room containing an unfamiliar mouse, a stranger, and the mouse being tested could move freely between them.
Now, a normal, typical mouse is curious, right?
Yeah.
It would go check out the stranger.
Exactly. A wild type or normal mouse spends much more time sniffing and interacting with the stranger mouse. We're social animals by and large. We're interested in others. But the knockout mice, the ones with the social deficits, they spent just as much time with the empty room as they did with the stranger mouse.
They just didn't seem to care about the social interaction.
They showed no preference. But then came the key part of the experiment.
They gave them oxytocin.
Well, they did even better. Using optogenetics, the same light-based technique we discussed, they specifically activated the oxytocin-producing neurons in these mice, and the result was immediate and dramatic. The mice suddenly spent significantly more time interacting with the stranger.
So simply tweaking the level of one hormone, one chemical, completely changed the fundamental social preference of the animal.
It did. It suggests that hormones like oxytocin don't necessarily create social behavior, but they act as a kind of social tuning knob for the brain. They prime the neural circuits to find social interaction more salient, more rewarding. It essentially changes the gain on the social signal.
So let's zoom all the way out. We have covered a lot of ground here. What does this all mean when we put it together?
I mean, we've traveled the entire biological hierarchy just like we set out to do. We started with a single cell, a physical structure, a neuron, described by Cautel, separated from its neighbors by a tiny gap.
Then we watch that cell generate an electrical spark, the action potential, by basically opening its doors and letting a river of sodium flood in.
We saw that spark travel to the end of the line and pop a chemical bubble to get its message across a synapse, turning the signal from an electrical spark into a chemical soup of neurotransmitters.
And finally, we saw how the whole system is regulated over long periods by these body-wide signals, these hormones in the blood, that change our very motivations and behaviors. Whether it's Bev smelling a childhood memory on an operating table,
Or a rat pressing a lever for a hit of cocaine.
or a mouse deciding to say hello to a stranger.
It all comes down to the machine. It's physical, it's
Yeah.
mechanical. The magic of our subjective experience is the mechanics of our biology. There is no software without the hardware.
And here's a final thought to leave you with, something to mull over. We talked about that dyadic fMRI, two people in two separate scanners looking at each other, interacting. If we are at the point where we can actually map the biology of interaction, where we can literally see the social brain lighting up in real time in response to another person, are we getting close to redefining what connection actually is? I mean, if I can see your brain changing my brain on a screen, does the barrier between me and you start to dissolve just a little bit?
That is a profound question. If all behavior is electrochemical, then a relationship is really just two electrical storms influencing each other across space. It's a beautiful, if slightly mechanical, way to view things like love and friendship, isn't it?
The shared electrical storm. I kind of like that. Thank you for listening to this deep dive into the hardware of the human mind.
So stay curious.
And a warm thank you from the Last Minute Lecture team. Good night.